Our lesson today will be about the light-dependent reaction. You know the photosynthesis process is divided into two main processes, light-dependent and the light-independent reactions. Both reactions take place in the chloroplast. Let's have a closer look for the structure of the chloroplast. Chloroplast is made of outer membrane and the inner membrane, and inside we have a liquid inside. This liquid we call it stroma, where the light-independent reactions take place because it has the enzymes, and the most important enzyme, Rubisco, which is the main drive for the light-independent reactions. Light-dependent reactions happen in membranes inside the chloroplast that we call them thylakoid membranes. And the collection of thylakoid membrane, we call them grana. The membrane, this is the outer chloroplast membrane, the outer thylakoid membrane, and there is a space inside, we call it thylakoid space. Some reactions take place in the membrane of the chloroplast, and the other reactions take place inside the thylakoid space. Both of them include only in the light-dependent reactions. Light-dependent reactions start when photons of light fall on the chlorophyll. It was found that the chlorophyll, where is the chlorophyll inside the chloroplast? Chlorophyll found inside the membranes of the thylakoid membrane. So this is the part of thylakoid membrane, and those circles, red circles, represent the chlorophyll molecules. Chlorophyll molecules are arranged in clusters or arrays. We call them light harvesting array or light harvesting clusters. There are two clusters here in the chloroplast, photosystem 2 and photosystem 1. It was found that the photosystem 2 is the main photosynthetic cluster, which is responsible for or initiate the light-dependent reactions. Light-dependent reactions start when the photons of light fall on the chlorophyll molecule inside the photosystem 2. As a result of that, chlorophyll has magnesium atom in the center. Magnesium is going to absorb the photons of light. The electron there will get excited from a lower energy level to a higher energy level. So we say that the chlorophyll has been activated. This process, we call it photoactivation. So when the photons fall in the chlorophyll becomes activated. Here the light energy is captured in the form of chemical potential energy. The electrons, the energy is going to pass from one chlorophyll molecule to the next within the photosystem 2 until it reaches to what we call reaction center. In the reaction center, there are chlorophyll molecules in the center. Those are considered as the last electron acceptor. So here, if two photons of light fall on the chlorophyll molecule, on one chlorophyll molecule, they are going to pass from one chlorophyll molecule to the next until they reach the reaction center. The two photons of light are able to let the chlorophyll to accept two electrons. Not any of those chlorophyll molecules can donate their electrons, but only the chlorophyll molecules in the reaction center. Those can donate their electrons, and we call them electron donor. They donate their electrons to a molecule here found in the reaction center. We call it plastoquinone. The plastoquinone, when it accepts the electron, electron accept, when the molecule accepts the electron, we say that it's reduced, so it will be a reduced plastoquinone. This reduced plastoquinone, it can carry a pair of electrons, and not only electron, also it carries lots of energy, and it drives the rest of the light-dependent reactions. And now you will see the role of the reduced plastoquinone to drive the rest of the light-dependent reactions. Here we have a closer look for the thylakoid membrane of the chloroplast. This is the thylakoid membrane. It's a phospholipid bilayer, completely similar to the structure of the cell membrane. And there are many molecules attached here. We have the photosystem 2 and the photosystem 1. Each of them has a reaction center with a pair of chlorophyll molecule in the center. Also, we have here a group of electron acceptor that drives the electron transport chain, and we have here the ATP synthase that drives the process of chemiosmosis or photophosphorylation. We said light falls, or the photons of light falls on the chlorophyll molecules. The chlorophyll will get activated by exciting of two electrons. This electron will pass from one chlorophyll molecule to the next until they reach the reaction center. The final molecule here is able to donate its electron to plastoquinine, so the plastoquinine will become reduced. Here, this process will make the chlorophyll molecule in the reaction center very good oxidizing agent, and here the area around it, or the chlorophyll molecule here in this area, is able to split water molecules into oxygen and hydrogen. The equation is 2H2O, releases 4 electrons, 4 protons, and O2. O2 will be given out and can get outside by diffusion as a byproduct of photosynthesis. While the main product of photosynthesis, which is sugar molecule, it has long processes in order to get produced. So here, as a result of splitting of water, also you can see that protons will be produced, and they will stay here in the thylakoid space, and while the electrons will go from one electron carrier to the next. So the electrons are going to move from through a group of electron carriers. This is why we call this process electron transport chain. And you know, as we have studied in the mitochondria, in the process of respiration, as electron pass from one electron carrier to the next, energy is being released. Here, the energy is used partially to split water molecules, and also part of energy is used to drive the machine that we are going to talk about it later on. So now you can see the electrons will pass from one electron carrier to the next until they reach to the reaction center of photosystem 1. Remember that also photosystem 1 absorb light energy, and the electrons will get activated. But the electrons here that get activated, they will move and be carried. The electrons will also move here from one chlorophyll molecule to the next, to the reaction center, and then to NADP, which is found here, to become reduced NADP. Because NADP is going to take the electrons along with the protons resulted from photolysis of water, forming reduced NADP. The reduced NADP will be used in the light-independent reaction, or what we call a Calvin cycle, that we are going to discuss it in the next lesson. Okay. What's left here as a result of build-up of hydrogen ions in this area, so the proton gradient will be established, because outside the thylakoid membrane there will be very low concentration of hydrogen ions. Now hydrogen ion concentration is going to increase in the space, so there will be a proton gradient built up. As a result of that, the ATP synthase can be driven now by the energy produced from the process, the electron transport chain, because as we said, as the electron pass from one molecule to the next, some energy is released. So this energy together with the protons are going to drive the ATP synthase machine or enzyme. The hydrogen here can pass now passively down their potential gradient, and in doing so the machine is going to rotate, releasing ATP from ADP and PI. Both of reduced NADP and ATP will be used in the next stage of photosynthesis, which is the light-independent reactions. Just I want to summarize the process and add some new terminology. Photophosphorylation, to remind you, photophosphorylation, which is the formation of ATP from ADP and PI. Where phosphorylation is taking place here, happen within the thylakoid membrane, where ATP synthase exists, and also, of course, it is influenced by light energy. This is why we call it photophosphorylation. Secondly, we said the reduced plastoquinine here produce the plastoquinine, which is the one which transfers its electron to the group of electron carriers, driving the rest of the process or the release of energy here. Thirdly, chemiosmosis, which is how the ATP synthase works to produce ATP. Look at this molecule here. This molecule, we call it plastocyanin. So I have here plastoquinine, and this is related to photosystem 2. So it accepts the electron from photosystem 2 and pass it through the group of electron carriers until it reaches to a molecule here. We call it plastocyanin. Plastocyanin is different from plastoquinine. Plastoquinine is hydrophobic, so it never gets removed or leave the thylakoid membrane, which is the phospholipid bilayer, and the hydrophobic. While the plastocyanin is hydrophilic, is water-soluble molecule, so this means that the electrons here can leave the thylakoid membrane and exist in the space, in the thylakoid space, because the molecule is soluble in water. So I can say that the plastocyanin is the final electron acceptor here, okay, and it's going to donate its electrons to photosystem 1 in order to replace the electrons that are gained by the NADP to get reduced. Okay. This is like a whole summary for the light-dependent reactions. Inshallah, we are going to continue the next video with the light-independent reactions, with more focusing on the role of reduced NADP and the ATP in the process. Thank you.